medical consults for them. So I guess that's the last thing I'll say about who I work for, and then we'll talk a little bit about the genes. So I know you guys had a homework assignment. What do you think of the video?
That's helpful.
In fact, I just posted the new one that has our updated genes. We just added 18 genes to the panel. So if you're interested in that, that was posted 2 weeks ago. You'll get to hear this fabulous voice for 45 minutes. All right, so this should be intuitive to everybody or anybody that's worked within psychiatry. So this basically tells you that what we're doing now, how we're treating our patients, treatment as usual leaves a lot of patients untreated, right? So here are the major NIMH studies. How much time do we have, by the way?
Oh, you have an hour and a half.
Okay. So these are the NIMH studies, you know, STAR-D, everybody knows what STAR-D is. STEP-BD was the bipolar trial. The Katie trial looked at schizophrenia. So these were the studies that had the best protocols in the world, some of the best clinicians in the world, and even in the best case scenarios, a third of your patients are left depressed. Bipolar disorder, at least 50% of bipolar patients relapse. Because when you're having a manic episode, who needs medicine? I'm not taking my lithium anymore, I feel great. Schizophrenia, 3/4 of your schizophrenic patients stop their first medication. So we need to do something different, and this is one of the tools in the arsenal that we hope sort of directs your medication therapy. So here's STAR-D in a nutshell, right? Everybody's familiar with STAR-D. If you failed your first trial, you are less and less and less likely to respond to future trials of medication. And it didn't matter what those second, third, and fourth trials were, whatever the medication was, if you were treatment resistant, you were less and less likely to fail. So less and less likely to succeed. So the moral of this story is let's select the right drug first, right? So if you select the correct drug, you have at least a 37% chance of remission. So let's look at your pharmacokinetics. Let's see how you break down medications, how you individually break down medications. Then let's look at your pharmacodynamics, your receptors, your enzyme systems, and let's see which classes of drugs are more or less likely to work for you. Because when you look in the literature, you're looking at averages, all right? And averages tell you a lot of information, but there's a lot of fuzz on those averages. So the goal here is to try to personalize therapy. All right, there's STAR-D, there's everything, all the drugs that we use in STAR-D, first step, second step, third step. And by the fourth step, they threw an MAOI in there, which are fabulous medications, right? Everybody loves MAOIs. And the MSAM patch isn't that bad. You guys like MSAM?
Yes.
Yes. Okay. So what do? We look at biomarkers, right? This is the whole goal here. Let's look at biomarkers. Let's see what that tells us. Let's see how predictive these are, and let's personalize your medication. Let's see if my laser works. Look at that, it works. All right, so specifically what I look at is SNPs, single nucleotide polymorphisms. We each have 3 billion base pairs. A change in a single one of those base pairs can change who you are, okay? I'll show you a real live example of this, the COMT gene, which you may have heard, be familiar with from the webinar. The COMT gene, a single change from a G to an A, can drastically change how you respond to things like Adderall. So here are the 18 genes that I look at. Can you guys see that? No, right? Yeah, so, well, don't worry about it. Anyway,
Maybe we should turn the lights down a little bit.
But no, this is all new. Dan got it for everything right here.
I'll call for help.
All right, let's see here.
Is this presentation something that you'll share with us eventually, or no?
This is, I made this specifically for you guys, and I'm probably going to use this internally, but potassium and sodium channels affect your drug therapy. Weight gain genes, atypicals are used for everything now. How can we isolate the patients who are most likely to gain weight from that? ADD, ADHD, the COMT gene and the ATRA2A gene, we put them together. So we try to make it user-friendly, so you look at different pathways of medications instead of just worrying about a diagnosis. Look at the underlying physiology behind it. And there are your gene frequencies. You have 2 seconds to memorize that. There you go. The interesting thing here is there's a lot of variation within different ethnicities. Take a look at the short gene here. So in Caucasians, it's about 20% have the short. That's the high risk for failure of SSRIs. But check it out in the Asian population, 70% have short. So, you know, same thing with MC4R. Pretty much all the genes, a lot of ethnic diversity with the frequencies. And this is some information we're going to capture on the front end of our testing from now on, because it's important to know your ethnicity, because it's going to impact your response to medications. BDNF is very interesting. So whether you're Asian or Caucasian, you have the opposite response to SSRIs and SNRIs based on your BDNF levels, and I'll talk a little bit more about that later. So there is your SLC6A4, your serotonin transporter gene. You notice that this one is long, notice that this one is short. That short genotype that I was talking about before, it's literally shorter. It's missing 44 base pairs. That 44 base pair deletion makes a dysfunctional gene. So I promised you I would talk about COMT and how a single base pair substitution results in real-world different proteins. So here's COMT MET, here's COMT VAL. Why do they call them MET and VAL? Because this single base pair substitution results in a methionine amino acid. But if you have a G here, it results in a valine amino acid. We could have just as easily called this an A or a G genotype, but in the literature they call it MET-VAL. They refer to the amino acid that's produced instead of the single base pair that's been substituted. Most of the other genes out there, they'll just tell you what base pair was substituted. Your genotype will be CT, AG, GG, whatever the case may be. For COMT and BDNF, they define them as VAL and MET. But that 1 out of 3 billion changes your phenotype. VALs respond a lot better to stimulant medications than MET-METs do.
Now, COMT is the enzyme that's responsible for the breakdown of dopamine.
Yeah, dopamine and norepinephrine.
Okay.
But the tricky part is you have more COMT in the prefrontal cortex than you do dopamine reuptake pumps. So COMT has a bigger impact on the PFC, bigger impact on cognition. Your VALs break down dopamine a lot faster than your MET-METs. So VALs are genetically low dopamine. How do you fix them? Boost dopamine. What's the easiest way to do that? Give them some Ritalin, give them some Adderall, give them some Wellbutrin. That all boosts dopamine.
Why do you say Val? Why do you say it?
So half your DNA is from mom, half your DNA is from dad. And when we talk about genotypes, it's that combination that makes your genotype. So every genotype you see here will have 2 alleles associated with it. And it's that combination of alleles that defines your genotype. So usually these genes are cumulative. So when I say short, that means mom gave you a short copy, dad gave you a short copy, and having 2 copies is worse than having just 1 copy.
Okay. So that would be a Val or a...
Yeah, there are VAL-METs also. Yeah,
Yeah.
so there's MET-METs, VAL-METs, and VAL-VALs.
And then there's short.
And there's long-short, short, or long.
But that's with a different gene.
That's a different
That's with
gene.
the
Yeah, no, that I see,
serotonin.
yeah.
Mhm.
Yeah, so that you can be heterozygote or homozygote.
Dan, originally, the literature I was looking at said that SAMI was recommended. And I saw in the new information that was eliminated. Why? Because I've had very good results with SAMI.
So this gets a little hairy, but
SAMI was
we'll go into it anyway.
recommended if you were a VAL.
The
I
other
mean,
way around.
I'm sorry,
MET-MET,
MET-MET,
right?
yeah.
So if you were COMT MET-MET, if you had high dopamine levels, we recommended SAMI because SAMI can help stabilize the COMT enzyme, which ideally would reduce dopamine levels. But at the same time, SAMI can methylate the COMT enzyme, which reduces the activity. So SAMI can really do 2 different things to the COMT enzyme, and it's not clear which direction it would go to.
Okay.
So
It's kind of the way
It's
I
like a
tested.
partial.
no, it's not a partial agonist like Abilify. It's just we don't know what pathway SAMI would be shunted to in the body. When you do it in vitro, when you just do it all in a test tube, SAMI stabilizes the COMT enzyme. When you do it in a real live human being, we're not sure what pathway it goes to. So just to play it safe, we decided to remove that recommendation.
Okay, thank you.
So, just a question, so you recommended there's someone who's Val, that they would respond well to Ritalin and Adderall,
Yeah.
and there was one other?
Oh, Wellbutrin. Yeah,
Oh,
Wellbutrin
and
is,
dopamines, okay.
Wellbutrin's basically baby Ritalin. It works the same way that Ritalin does, it has a weaker binding affinity to the dopamine pump.
So if people just take amino acids like as a supplement, does that help or does it not make a difference?
Good
Like
question.
if you took Valine as like an amino acid supplement, would that make
Yeah.
any difference?
Possibly, there's just no data to support that. And, you know, so we have a lot of integrative clinicians which will do blood levels on amino acids and whatever the case may be. And I don't know how predictive that is because it doesn't tell you what's getting into the blood-brain
That's
barrier.
what I was thinking, yeah.
So, good question, I don't know the answer to it.
Yeah. So Val-Met is normal,
Val-Met's
right?
normal, so
Val is
yeah,
low,
Goldilocks.
Met is high.
Val-Met in the middle, Met high dopamine,
Okay.
Val low dopamine.
So what do you do, I mean, Val was what came back in most of my tests, but what do you do if somebody's Met?
Good question. So a meta-analysis just came out about 3 months ago that showed if you were Met, you had a better response to atypical antipsychotics. If you think about it, it makes sense. If you're high dopamine,
Yeah,
you want to
yeah,
block
yeah.
that dopamine. Atypicals do exactly that. It's possible that may be added to our report soon. I'd like to see, there was a Chinese study that supported that and an American study that supported that. I'd like to see some more data. It was only tested in schizophrenics. I'd like to see how effective that is for augmentation of antidepressants also, if it holds true there.
Yeah. What journal is this data coming out in?
Oh my God, a million of them.
Oh, okay,
There's
so
a
it's-
pharmacogenomics journal, there's genetics and psychiatry, it's all over the place.
Okay.
If you do your testing, does it come with some kind of like manual to interpret it?
Yeah, so there's a
Yeah.
report interpretation guide that we give you.
Okay.
It's pretty self-explanatory, the report, so it gives you the gene results, and then it lists the top 100 medications you would use in psychiatry, and how those genes affect each one of those drugs.
And for the first 3 that I did, you can call, and I don't know who I spoke to. Was it you?
Probably.
Oh my God. I was going to say the person was wonderful. Actually, it was really the first one. The second and third, I had more questions about administrative stuff and billing and Medicaid and that sort of thing. Oh, before I forget, Medicaid is not covered in New Jersey, but it is in Pennsylvania. How about New York?
Not yet.
Okay.
So
Are
we have
you guys
Medicare.
trying to get
Yeah,
that?
we have our applications into every state.
Okay.
New Jersey's an oddball. We had to have Pennsylvania Medicaid.
It's called Chris.
Yeah, before we had New Jersey Medicaid. New York is in the works, but that's pending. We have 18 states Medicaid so far, so.
How we
we'll
go with
get there.
commercial insurance in New York?
It's hit or miss, yeah, so, some don't pay anything, some pay great, everyone else is somewhere in the middle, where, you know, we just expanded our I guess our third-party department to try to get more contracts, and that's